it's gone off. And I especially look at meat that are in little packs, little packets, where you kind of take out the oxygen. There's no oxygen in there, it's flat. So when I mean spoilage, I mean that the flat pack begins to kind of rise into a little football. And I basically look at what happens inside. Ireland's main income is through agriculture and food production. And red meat is one of Ireland's main exports. So you send the meat outside of Ireland to other overseas markets. So if there is spoiled meat, if you have gone off meat which you cannot send away, then you have a problem. You economically have a problem where it will hamper the meat industry. So my aim is to try and find out why it's happening and what's causing it and what I can do to help the meat industry reduce it or prevent it altogether. So at the moment, as I said before, I'm working on trying to control the occurrence of this kind of spoilage in meat. So at the moment, I'm working with some chemicals that could potentially kill the bacteria or kill the bugs I work with, or the bugs that cause this spoilage. So at the moment I'm just trying out a range of different chemicals to see at what time point or how many hours does it take or how many minutes does it take for these agents to kill the spores. So if they are present where they, at meat production, at meat plants or where they produce meat, or where they package the meat, then we could potentially use these agents or these chemicals to reduce the growth or to prevent the growth and control it eventually. Starting an experiment today, I had to obviously get a lot of prep work done. So if you see here, I have my anaerobic boxes. This is the box I use to put my plates in. So if you can see here, I'll try to take one for you. You can see that there are plates here. So I basically grow my bacteria on these plates. They're Columbia blood agar plates. So blood is it's a very highly nutritious medium for the bacteria I grow. And I have my tubes here where I'll be putting my sporicidal, my chemical agents to look at their sporicidal effects or bactericidal effects on the bacteria. But I have my tubes ready, my boxes ready. So yeah, pretty much I think the main thing is to kind of get organized when you're doing an experiment, because you don't want to come in on the day and things are all over the place. I usually like having everything in order and everything good to go. So when I do start, probably in a couple of hours, I'll have everything at hand. I don't want to move around too much. Show you. The anaerobic cabinet. So this is where I usually do most of my work. So as I mentioned earlier, that the packaging that the meat goes into is an anaerobic environment where the oxygen is, there's no oxygen in there, so that there's CO2 flushed in. So this cabinet here makes sure that there is no oxygen. So I usually put my plates or my samples in here to make and keep them overnight to make sure that they're deoxygenated. So it'll be more favorable medium for the bacteria to grow in and favor their growth. So vacuum packaging, as it's called, is an anaerobic environment. So it actively makes sure that there's no oxygen, because in meats, usually, if meat is exposed to oxygen, there is a lot of aerobic spoilage, where you have bacteria that grow well in an atmosphere where there is oxygen. But my bacteria grow in an environment where there is no carbon dioxide, and they also produce carbon dioxide as a result of fermentation in the vacuum packaging. I studied biomolecular science and biosciences in college, so we did a lot of modules involving microbiology. And when I finished my course, I always wanted to do a PhD, and I think it came from when I did my research project in my 4th year of college, where I could independently work around on my own, direct my own work. So I always wanted to do a PhD and see where I could go with it. And I was looking at PhDs in microbiology, immunology, molecular biology, and then this opportunity showed up, and it was a very interesting project where there is a very structured project where you could see why you're doing it and where you're going with it. And I also liked, I heard a lot of good things about Teagasc and the people here. So yeah, I think that was pretty much it. And then I'm quite happy with the experience I've had so far with Teagasc. You need to be 100% confident that you really want to do it, because it is a long time for life. It is about 4 to 5 years of your life that you will spend doing a PhD. So you need to find a project that really suits you, suits your skill set, suits your practical and your theoretical knowledge that you've gained from college. So really be into the project you're doing. I really love the area you're working in, because it really does help and really does add to your whole experience if you really love what you're doing. And I think just be really confident, believe in yourself that you can actually do it, because it is a big commitment. As my supervisor once said to me, it's not a holiday. You have to put in every minute into it, and it's all about hard work, and it's all about how you drive your project forward and how you finish it, because at the end of the day, you're getting a PhD out of it. So you have to be committed, you have to be confident, believe in yourself, and just really enjoy it, I suppose. Just be really in for the ride, I suppose.